So the second explanation regarding the action of this RHO describes that RHO pulls RNA out of the polymerase, resulting in termination. So this is complex, and RNA transcript is exiting this polymerase, this complex. So this RHO pulls this RNA out of this complex. So in this way, the transcription stops. This is a second explanation. The third explanation describes that RHO induces a conformational change in the polymerase, and this causes the enzyme to terminate the transcription. Most recent experiments suggest that the last of the above three explanations may be correct, and that is which involve the conformational change in the RNA polymerase, and that conformational change causes the elongating complex to stop, and this stoppage then follows the dissociation of this complex slowly. Recent studies also have suggested that RHO binds to RNA polymerase throughout the transcription cycle. So it is that this RHO has already been attached to this complex. Thus, RHO doesn't reach polymerase by translocating along a nascent rut-containing transcript like TRCF. So it doesn't work like TRCF. That is, it binds to DNA, then it translocates and then pushes the polymerase forward. So this is not true. Rather, RHO binds polymerase early in the transcription, and then at some point also binds the RNA transcript. So it has already bound to the DNA, and then during transcription it also binds to RNA transcript, which is being exuded from that of elongating enzyme. The role of translocation by RHO is thus perhaps to tighten the resulting RNA loop, and when sufficiently tight, polymerase elongation ceases. So the purpose of this translocation of this RHO factor, if it is already attached to this complex, the purpose of translocation of this RHO is just to tighten up this loop of RNA which is being synthesized. So when it is sufficiently tight, the RNA polymerase stops transcription. Then how is RHO directed to work on particular RNA transcript? So because there are many RNAs present in the cell, how this RHO will only work on particular or specific RNA transcript? First, there is some specificity in the sites it binds, that is the rut sites. They are specific for the binding of RHO. These sites consist of stretches of about 40 nucleotides that do not fold into a secondary structure. So there is a sequence of about 40 nucleotides, and these 40 nucleotides, they remain in primary structure, that is, they are not double-stranded, rather they are single-stranded. So the RHO binds to these sites. They are also rich in cytosine residues. So these sites are also rich in cytosine residues. The second level of specificity is that RHO fails to bind any transcript that is being translated. So there may be some transcript present in the cell, and those transcripts are being translated for the synthesis of proteins. So this RHO will never bind to those transcripts which are in the process of translation. in bacteria, transcription and translation are tightly coupled. So translation initiates on growing RNA transcripts as soon as they start exiting polymerase while they are still being synthesized. So in bacteria, because there is no well-defined nucleus, so this transcription machinery is present in the cytoplasm, and when this transcription starts and a new mRNA is synthesized, so as soon as it exits the polymerase, it start exiting polymerase, the translation may start. Normally translation start. So these translation and transcription are coupled in bacteria. Thus, RHO typically terminates only those transcripts which are still being transcribed beyond the end of gene or operon. So that transcript which is being simultaneously transcribed and translated, so that transcript would be still in contact with that polymerase complex, and that will be in the process of transcription. So this RHO will only stop this mRNA transcript, and it will not bind to any other RNA transcript. This is RHO, and this one is a RUT site. So it binds here, then it translocates, and it reaches the RNA polymerase. This is RNA polymerase, and then it stops transcription. In a second explanation, this is RHO, and here it is RUT site, and it binds here, and then it translocates here and strikes the polymerase. And in another illustration, you can see here it is RHO, which contains 6 subunits, and the RNA is looped around this. So in this case, it tightens the loop and stops the termination.